Okay, so good evening, everyone. So I welcome you all to the week 5 problem solving session of this NPTEL course, Genetic Engineering Theory and Application. Okay, so I hope all of you have gone through the week 5 lectures. So till now, we have learned a couple of different concepts starting from basics of cell biology to biochemistry. Then we learned about basics of cloning, what are uh, what are the different enzymes which are required in cloning. Then we saw what is PCR, what are the steps in a PCR reaction. Um, then genomic library, cDNA library, how to screen the different libraries. So we looked all into all of these concepts. So in week 5, the main concepts which were discussed, can anyone recall what all topics have been covered in week 5? Okay, so I'll start today's session now. Um, okay, hello everyone. I'm Akash Jyoti Pathak, and I'm one of the PMRFTAs for this NPTEL course, Genetic Engineering Theory and Application. This course is being run by Professor Vishal Trivedi of IIT Guwahati. Today we will solve a few questions which will help you to solve the week 5 assignments, and also we will quickly have a quick recap to brush up the concepts which were covered in week 5. Uh, so, so let's start today's session. So, in between, if any one of you have any doubts, or if you but if you are not able to understand something, then you can write it in the chat, or you can ask me by unmuting yourself. Okay. Okay. There were a couple of important concepts covered in week 5. So in week 5, we saw that we have started the recombinant DNA technology. So there are two parts. So week 5 deals with the part 1 of the recombinant DNA technology. So in the previous lectures, we learned about how we can perform cloning. So how we can clone our gene of interest into a vector. So now after we have cloned our gene of interest, so once we have cloned the gene of interest into the vector, now what is the next step? So we have cloned the gene of interest into inside our vector, and we have done PCR reaction to confirm whether my clone gene of interest is present, or we have also done restriction digestion and we have checked, yes, my gene of interest is present, and we have also sent our plasmid for sequencing, and we found that yes, my gene of interest is present in my vector. Now, once this gene of inter we have the recombinant DNA. So the clone gene of interest, when it is present inside the vector, we call it recombinant DNA, isn't it? So this is the recombinant DNA. So once we have prepared the recombinant DNA, the next step will be what? Next step will be to introduce it into the host cell. Okay. So step number two is to introduce the recombinant DNA, rDNA, into the host cell. In the last session, we saw what are the different vectors that we can use for cloning our gene of interest. So depending on the size of our gene of interest, we have different types of vectors. So there are mammalian vectors, then there are baculovirus-based vectors, then there are bacteriophage vectors. So different types of vectors we learned in the last lecture. So now we have the recombinant DNA, and today what we will understand is how we can introduce the recombinant DNA into the host cell. So there can be different types of host cells. So depending on our experiment, we can use bacterial host cell, we can use yeast host cell, isn't it? We can use mammalian host cells. So what are the various methods by which we can introduce the recombinant DNA into these different types of host cells that we will learn today. We will quickly have a recap of what all was covered, and then once this is introduced into the host cell, then again we have to screen the host cell, whether the host cell screen the host cells. So there will be some host cells which will take up the recombinant DNA, and other host cells which will not take up the recombinant DNA, or and it will take up the the DNA which does not have our gene of interest. So again, we need to screen for the host cells. Okay, and then once all this is done, then what is done is actually the plasmid is isolated from the host cell, and sequencing is done so that we can be confirmed that my plasmid has the gene of interest cloned inside it. Okay, so we will also learn about the two most widely used sequencing method. One is the Sanger sequencing, and another which is not used nowadays, but it was used earlier, which is Maxim Gilbert sequencing. Okay, so the topics which were covered is that the intro how we can introduce the cloned gene into the host cell. So there are a couple of methods, starting from the heat shock method, which is regularly done for transforming our recombinant DNA into the bacterial cell. Then then another is the electroporation method. Okay, so an electric field or yeah, 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 yeah. Electric field is given to the bacteria to create pores in the membrane so that the plasmid can be taken up by the cell. Okay, then how transformation is done in yeast cells. So in yeast cell also we will see 2 to 3 methods by which we can introduce or put the clone gene of interest into the yeast cell, and how and the next, what we will see is how we can introduce the recombinant DNA in mammalian cells. So this process is called transfection. So when we are introducing recombinant DNA in mammalian cells, that process is called transfection. While the same thing when we are doing it in bacterial cell, that process is called transformation. Now, once these are done, then next step, as I was explaining, that we will screen for the host cell which contains the recombinant plasmid. So there are a few methods by which we can screen the hosts which contain the recombinant plasmid. So insertional inactivation, and another is a blue-white screening. Then we'll quickly cover the DNA sequencing topic. So Sanger sequencing and Maxim Gilbert. sequencing. Okay, so let's start with the how we can introduce the gene of interest or how we can introduce the recombinant DNA into our host cell. So if you see this figure, it is a summary of what all we have seen till now, or the entire cloning, gene cloning methodology. Okay, so entire gene cloning, it is a summary of this is a figure showing the summary of gene cloning. So what you can see here, so you have a source DNA, and in that source DNA, this is the target gene or the target DNA that you want to clone it inside the vector. So this is your cloning vector, and this is your target DNA. So you do enzymatic fragmentation. We saw how we can from the genomic DNA, how we can get how we can isolate the genomic DNA and how we can get the target DNA from the genomic DNA. So we covered these topics in the earlier sessions. So by enzymatic fragmentation, we got the target DNA and we got the vector DNA. And then what is done? Ligation is done, isn't it? So ligation is done, and we clone the target DNA in within the vector DNA. And then today what we are seeing, so till all these things we had covered already in week 1, 2, 3, and 4. Till week 4, we have covered all these topics. Now today what we will mainly deal with is how this DNA, recombinant DNA is introduced into the host cells. And so you can see here in this figure, so we have this is the recombinant DNA, and this recombinant DNA is inserted inside the host cell. And then after this recombinant DNA is inserted into the host cell, then we can express the gene which has been cloned. So how we can express the genes by and produce our required protein of interest. Okay, protein encoded by clone gene. So we will see these two things: introduce DNA into host cells and isolate the cells with cloned genes. So screening, how we can screen for the recombinant for the host cells which contain our recombinant DNA, and how first how this recombinant DNA is introduced into the host cell, that we will see here. So let's start with Griffith's experiment, because this tra this that how we introduce the recombinant DNA into the host cell. That comes from the Griffith's experiment, who first discovered that bacteria have the ability to take up DNA from the environment. Okay, so we all have learned about Griffith's experiment in class 11th and 12th. So what did Griffith do? So you can see here that in the this is an experimenter. So Griffith, what he took two different types of bacteria, right? Type 3 S bacteria. So his model organism was what? It was Streptococcus pneumoniae, isn't it? So it was Streptococcus pneumoniae. Streptococcus pneumoniae. And so there are two different strains of the Streptococcus pneumoniae. One is the S3 strain and another is the R2 strain. So the S3 strain is what? So this is the smooth strain. This is the smooth strain. Why? Because this type of this strain has a glycocalyx covering over its cell wall, and that is why the colonies appear shiny. And the rough strain do not have this glycocalyx covering, so they appear rough. And because of this covering, this coating, it makes the S3 strain virulent, while the R2 strain is virulent. Virulent means it is pathogenic. So the S3 strain has the ability to cause pneumonia infection. Okay. S3 strain has the ability to cause pneumonia infection, so it is pathogenic. Okay, so what he did, he cultured the S3 strain. So you can see this red colored uh, col- uh, bacteria that has been shown in this cartoon representation. So this red colored bacteria, these are the S3 type, and this blue colored are the R2 type. So R2 type is non-virulent and the S3 type is virulent. And then he cultured these bacteria and injected the S3 bacteria into a mouse. And he also injected the R2 type uh, bacteria into the mice. And what he found that the mice which were injected with the S3 bacteria died, while the mice in which the R2 strain of bacteria was injected, they were alive. Now, in the next step, what he did, he heat killed the S3 type. Okay, so the S3 type is virulent. It is causing pneumonia infection, and as a result, the mice is it is causing the death of the mice. Now, if heat-killed S3 type bacteria is injected into mice because this bacteria has been heat-killed, so it has lost its pathogenicity, and as a result, the mouse lives now. Okay. Now, what he did, he mixed the heat-killed S3 type bacteria with the virulent R3 strain of bacteria, and what he found that what was the expected result? The expected result should have been that because this S3 type bacteria is heat killed and the R2 cannot and it has lost its pathogenicity. So if I heat killed the S3 type, then it can no longer cause any disease, and the R2 does not cause any pathogenicity. So the mice should have stayed alive, isn't it? But what he observed that the mice died as a result of injection of this heat killed mixture of this heat killed S3 and R2 bacteria. When these two were mixed and injected into the mice died. And when the blood was isolated from the mice, it was what was found. So from the blood, he could isolate type S3 virulent bacteria. So the when the blood from this dead mice was isolated, he found that in the blood there are S3 type virulent bacteria present. Okay, so from all these experiments, what he concluded that a substance in the heat-killed virulent bacteria genetically transformed the type R2 bacteria into live virulent type S3 bacteria. So he concluded that that there must be some substance in the S3 type of virulent bacteria, and this substance converted the R2 strain into the virulent S3 strain. Okay, so that was his conclusion, and as a result, the mice died. Okay, so this was Griffith's experiment. So Griffith observed that small amount of living type S3 bacteria injected into the mice caused the mice to develop pneumonia and die. Okay, so the S3 bacteria, when injected into mice, it will die. When the R2 type bacteria is injected into mice, it remains alive. Okay, because this is non-pathogenic strain of the bacteria. And when large amounts of heat kill type S3 bacteria was injected into the mice, then also the mice remained alive because the bacteria has lost its we have killed the bacteria. And but when unexpectedly, what was found that when the heat killed S3 type was mixed with the R2 type, heat killed S3 mixed with R2, then the mice died. Okay. So from here, so actually what happened? So after then a couple of other discoveries took place one by one, and what was found that it was actually the DNA from the S3 bacteria which was taken up, which was taken up by the R2 bacteria. And as a result, the R2 bacteria now became pathogenic, and R2 bacteria was converted into the S3 type of bacteria. Okay. So that is what, so that is this process. This process was given the name as transformation. So any bacteria has the ability to take up the DNA present in the surrounding. Okay, so that is what is. So this was covered in the lecture videos as in the lecture classes as well. So in the lecture, this topic was covered, where you can see that the same thing has been shown over here as well. So that this is now what is you done in the laboratory. Okay, so this the same thing that we saw in the experiment, that one bacteria has the ability to take up DNA from the surrounding. The same thing is done in the laboratory. So in this figure you can see that the plasmid from one type of bacteria is isolated, and then this plasmid is added to the recipient cell. Now this recipient cell can take up this plasmid DNA. Okay, from the surrounding, it can take up the plasmid DNA and it can integrate the DNA in its own genome. Okay, or if it is a DNA, then it can, by the process of homologous recombination, it can integrate the DNA into its own genome. Or else, what it can do, if we have put the plasmid DNA, um, put the plasmid DNA into the competent cells, and we have taken the competent cells and we have added the plasmid DNA, then this plasmid can also be taken up by the bacteria. Okay, so we will see what are competent cells and other stuff. So this is what is called transformation. So once the gene of interest is cloned into a vector, okay. So once the gene of interest is cloned into a vector, the construct can be put back into the bacterial cell through a process called transformation. So most type of bacteria, including E. coli, take up only limited amounts of DNA under normal circumstances. Okay, so what has been seen is that if you take a normal bacteria and if you just add DNA or plasmid DNA in the media, say for you have added the plasmid DNA in the media, then there is a very less chance that this bacteria will take up the plasmid DNA. Okay, so that is what is explained over here. So take up only limited amounts of DNA under normal circumstances. It will not take up the DNA. So to transform these species efficiently, the bacteria must undergo some physical and or chemical treatment that enhances their ability to take up DNA. So cells that have undergone this treatment are said to be competent. And so, normally in normal situation, the bacteria will not take up the plasmid DNA on its own. It has very less tendency to take up the DNA. So if we want the bacteria to take up that DNA, then we have to do some chemical and physical treatment. We have to do must the bacteria must undergo some physical and chemical treatment so that it can easily take up the DNA from the environment. Okay, and how this is done? So for E. coli cells, what is usually done is that the bacteria is treated with calcium chloride, 50 millimolar calcium chloride, and this treatment of bacterial cells with calcium chloride makes the bacterial cell competent. Now, there is some pores are created in the bacterial membrane, or there is some um, this calcium ion discharges because calcium is a positively charged, and it will the plasmid gets can be easily taken up by the bacteria now. Okay. So now we will see what are the different steps of transforming a recombinant DNA into a bacterial cell. Okay, so what is done? First of all, what is actually done is we take we will first have the LB media, isn't it? 200 ml LB media. We can autoclave it, and then we can inoculate the bacteria in this LB media, and then we will allow the where then we will incubate it at 37 degrees Celsius and allow the culture to grow. So once the culture has grown and we get an OD of around 0.4 to 0.6, or not 0.6, 0.2 to 0.4 at this stage. So at the mid log phase, what is done? The culture is harvested. The culture is harvested, and then the pellet that we get, this pellet is then resuspended, and there are a couple of steps uh, uh, by which the cells are made competent. So basically, the main thing what we are doing is we are taking this pellet and we are treating it with ice cold calcium chloride. while so I'm not going to that steps one by one. So that you pellet it down, and then you add calcium chloride, then you keep it on ice for some time, and then you again spin it and discard the supernatant. So there are many steps, but essentially what is done is the bacteria is treated with this calcium chloride, and then once this makes the cells competent. So once the bacteria is treated with calcium chloride, we get competent cells. So these are competent cells. Now the next step is what? So once we get the competent cells, then this competent cells, the plasmid DNA is added. Okay, plasmid DNA is added, and then we go for the, so now we want this plasmid DNA to enter inside the competent cells. So for introduction of the plasmid DNA into the competent cells, we do a heat shock method. Okay, heat shock method. Okay, first we take the competent cells. Okay, so around 100 microliter of the competent cells are taken, and then 100 nanogram, around 100 nanogram of plasmid DNA is taken, and then it is added to the competent cells, and it is incubated on ice for some time, for around 15 to 20 minutes, and then a heat shock is given. So this this microcentrifuge tube is then taken in a water bath, which is set at 42 degrees Celsius, and around 1.5 minutes, around 90 seconds or 1.5 minutes, this heat shock is given. And what happens when this heat shock is given, this treatment creates transient openings in the cell wall that enable DNA molecules to enter the cytoplasm. Okay. Now, after this heat shock for 90 seconds, the bacteria is immediately placed in the ice bucket. It's placed in the ice bucket for 2 minutes, or you can keep it for 15 to 20 minutes. Okay. And then after this step, what is done? After this step, LB media is added around 8. So if you have taken 100 microliter of competent cells, okay, so this there was a video in the lectures also, how this is done. So it was demonstrated to you how this heat shock method is done in the laboratory. So if you have taken 100 microliter of competent cells, you will add 900 microliter of LB media, and then you will allow this. So this is called the recovery media, and then you will allow incubate it at 37 degrees Celsius for 45 minutes to 1 hour, and then after this you again spin it, and the pellet is then plated, or you use a spreader and you plate it on LB agar plates. Okay. And then once you have plated it, you will incubate it again at 37 degrees Celsius, and the next day will come, and you will observe if you have got any colonies or not. Okay. So that is the entire process. So what are the steps? First of all, you need to prepare competent cells. How do you prepare competent cells? By treating the bacterial cell with calcium chloride. Okay. Then the next step is what? Next step is, once you have prepared the competent cells, you will give a heat shock. Heat shock is given at what temperature? It is given at 42 degrees Celsius for 90 seconds. Okay. Then after the heat shock, you again incubate it in ice for 5 to 15 to 20 minutes. Okay. And then you add the recovery media. Recovery media is basically your LB media. You pour the LB media and you again incubate it at 37 degrees Celsius. And after all these steps, what you do? You plate it on LB agar. Plate it on LB agar. Okay. So you can see this figure here also. So the E. coli cells in log phase of growth, they are transferred to poured calcium chloride solution. Okay. Now, this ampicillin-resistant plasmids are added to the experimental cells only. So once you add it to a whole calcium chloride, you have prepared the competent cells. Now, once you have prepared the competent cells, you add the plasmid. So the plasmid, if you are plating it on the LBV agar, which is having ampicillin-resistant, or you have poured ampicillin, supplemented the media with ampicillin, then you will add the ampicillin. So you'll add the ampicillin resistant plasmids, and then in the next step, you give a heat shock at 42 degrees Celsius. Then again, you place it on ice, and then you do the recovery at the recovery media, and after that, you plate it. Okay. So these are the steps. Okay. Now, what are the now there are different chemical by which you can make the cells competent. So for E. coli cells, what chemical we use? We use calcium chloride. So divalent cation salts are usually used, like calcium chloride, rubidium chloride also can be used. Okay, but there are for different bacterial strains, different chemical or competent agents are used. So this table has been taken from the lecture slides. So you go through this table nicely. So this Streptococcus pneumonia, what competent agent is used? It is mitomycin C and fluoroquinolone. Okay, for Bacillus subtilis, how is it made competent? It is made competent by treating it with UV light. For Helicobacter pylori, what is added? Ciprofloxacin. For Legionella pneumophila, what competent agent is used? Mitomycin C, norfloxacin, ofloxacin, nalidixic acid, bicyclomycin, hydroxyurea, UV light. Okay. So these are some things you need to remember. These points you need to remember, that for which bacterial strain, what competent agent or chemical agent you have to use. Okay. So the role of calcium chloride, so if you have understood, answer this question quickly. So what is the role of calcium chloride in preparing competent cells? Option A is it provides nutrient to the bacteria. It helps in uptake of DNA by swelling the cell wall. It acts as a selective agent for antibiotic resistance and it kills the non-competent cells. What will be the answer? So just now we learned that it helps in uptake of DNA by swelling the cell wall. Okay. So it will help in the uptake, but the DNA will only be uptaken when you follow the heat shock procedure. Okay, so if you do not give the heat shock and if you just add the plasmid to the competent cells, then the plasmid will not be taken up by the competent cell. Okay, you after adding the plasmid, you have to give a heat shock. You have to put it in 42 degrees Celsius, then you have to put it in the ice and then add the recovery. You have to follow this process. So your calcium chloride is only what it is doing. It is only helping in the uptake of DNA by swelling the cell wall. It is not it is not helping or the calcium chloride is not causing the DNA to be uptaken by the cells. For that, you have to do the you have to give it a heat shock. Okay. So yes, answer is option B. So this is again from the lecture video. So this topic was covered, right? So the bacteria is incubated with divalent cation like calcium chloride, magnesium chloride, or manganese chloride. Manganese chloride or rubidium chloride for 30 minutes at 4 degrees Celsius. And what happens during this process? The cell wall of the bacteria gets swollen and it gathers factors which is required for the intake of DNA docked on the plasma membrane. Okay, so once you treat the bacteria with calcium chloride, then the plasmid gets attached to the cell wall. Okay, the plasmid gets attached to the cell wall, but once you give the heat shock, then this plasmid you give it a heat and then yeah, then quickly you place it in ice. So that is the heat shock. And then, so what happens now? After this, the plasmid gets introduced or is uptaken by the cell. Okay. Now, following agent that can make Streptococcus pneumonia competent is, so just now I was showing you the table. Okay, so you go through the table nicely. So for Streptococcus pneumonia, the competent agent is mitomycin C. Okay, it is mitomycin C. So you follow the table that I have added in the previous one or two slide. Okay. Now during transformation, the cells were moved to chilled medium after the heat shock. So after you give the heat shock, you quickly place the cells in the ice or in the chilled medium. Why is it so? To halt cell division, to seize DNA integration, speed up recovery of transformed cells, and kill non-transformed cells. So what is the answer? So basically, when you are preparing competent cells, you have to be very careful. You have to be very careful. Why? Because your competent cells are very prone to. So once you have treated calcium chloride, you have treated the cells with calcium chloride, then these cells have a tendency to lyse. They become very fragile, and if you do not handle it carefully, then the cells can lyse. Okay, so when you are pipetting in and you are adding the different components, you cannot pipette it vigorously. You have to pipette it very slowly, and because the cells have become very fragile, and after that you are again giving it a heat shock, and so that at all these steps, so when you are doing this, you do not do carefully, then your competent cells may be lysed. So after this, what is done after the heat shock? The cell is quickly placed in the ice for the to speed up recovery of the transformed cells. Okay. So the answer is option C. And after this step also, we add the LB media. We again add 900 microliter of LB media, and we incubate it for 45 minutes to 1 hour, so that the cell wall that because when you are giving the heat shock, the cell wall and everything that gets there are pores which are created on the cell wall and cell membrane. So to so that the bacteria can recover from this extensive chemical and this physical treatment. The recovery media is added, and also after the heat shock, we quickly place it in ice for the recovery of the transform cells. Okay, so answer is the option C. So this question is very logical. If you go through the options also, you can easily eliminate and answer, because halt cell division, why will it halt cell division? So that has no logic that as to why it will halt cell division, and sees DNA integration. So this is also wrong. You can eliminate an answer as well. Okay, and it will not kill non-transform cells. Why will it go to kill the non-transform cell? You have not added any. Yeah, you are not plating it on. So once you plate it on the LB agar media, so only those cells which have the plasmid containing your recombinant, containing your antibiotic resistant gene, only those will survive while the others will get killed. But here you are not plating it on LB agar media. You are just placing it on ice, so that will not kill the non-transform cells. Okay, so answer is C. Now, transformation of yeast cell, how we can introduce the recombinant DNA in yeast cell. So one of the method is lithium acetate, PEG, and single-stranded DNA. So this method, what is done? It combines. So these things are, you need to yeah, you need to remember a few points, like how what are the methods that can be used for transformation of yeast cell, and what are the methods that can be used to transform a bacterial cell. Another method is electroporation, that was already covered. I am not so that is that you can if you have gone through the videos, you must have understood. But this method, so lithium acetate method, what it is done is it combines lithium acetate treatment, PEG mediated membrane effects, and carrier single stranded DNA to maximize the transformation efficiency. So why is lithium acetate added? So the lithium ions increase each cell membrane permeability by destabilizing the cell wall and membrane, facilitating DNA uptake. So in case of bacterial cell, we were treating with calcium chloride. Now here what we are doing for each cell, we are treating it with lithium acetate. Now, what the lithium ions are doing is it is making the cell membrane permeable, and also it is destabilizing the cell wall and cell membrane. And in the next step, the polyethylene glycol is added. So this polyethylene glycol acts as a crowding agent that induces close contact between DNA molecules. So actually, before this step, the single-stranded DNA is added. Okay, the single-stranded DNA is added. Why? Because this single-stranded DNA is prevents non-specific uptake, or no, it blocks the non-specific sites on the cell wall and makes the plasmid available for uptake. So after you have added lithium acetate, treated with lithium acetate, next you add single-stranded DNA. So these are also called the carrier single-stranded DNA, usually salmon sperm DNA. So it serves mainly three functions. It protects the transforming DNA from nucleases. Okay, it enhances the association of DNA with the yeast cell surface by acting as a bridge or competitor, improving transformation efficiency. And the most important point is that it blocks the non-specific sites on cell wall. The non-specific sites will be blocked, and it, as a result, it will make the plasmid available for uptake. Okay, and the last is that we add polyethylene glycol. So the polyethylene glycol, what it does, it acts as crowding agent and it induces close contact between DNA molecules and the yeast cell surface, promoting membrane fusion and endocytosis like uptake of DNA. So in this in this slide, what all you have to remember, you have to understand that what is the function of each of the components which are added during transformation of each cell. So what is your lithium acetate doing? Your lithium acetate is mainly making the cell wall permeable, or it is permeabilizing the cell membrane so that the foreign DNA can be uptaken by the cell. Next, what is your single-stranded DNA doing? Single-stranded or carrier DNA doing? It is blocking the non-specific sites. Okay, it is blocking the non-specific sites on the bacterial cell surface. Sorry, not bacterial. You are doing yeast cell surface. Okay, blocking the non-specific association, non-specific sites. And the last is your PEG. PEG, what it is doing, it is the crowding agent. This PEG is actually very viscous, and this PEG is basically it is allowing the yeast cells to come in close contact with the foreign DNA so that it can be easily uptaken. Okay. So it induces close contact. It induces close contact between the DNA molecules and the yeast cell surface. And it will also promote membrane fusion and endocytosis like uptake of DNA. So understand the different yeah, chemicals that are used and what is their function. Okay. That is the thing you have to remember over here. So now answer this question. So we discussed just now. So what was the purpose of adding single-stranded carrier DNA in yeast transformation? So option A is block non-specific sites on the cell wall. To block non-specific sites on the cell wall. Option B is to provide energy for the cells. To increase plasmid replication to enhance cell growth. So just now we learned the answer is option A, isn't it? So it will block the non-specific sites on the cell wall, and the cell can easily uptake the foreign DNA, the plasmid or the recombinant DNA can be easily uptaken by the yeast. Okay. So, so yes, it was explained in the lectures as well. So in this method, what is done? The yeast cells are incubated with a transformation mixture of lithium acetate PEG 3500. So there are different PEG 4000, PEG 3500, PEG 8000, different type of PEG are there in the lab. And then another thing what is added is single-stranded DNA carrier, single-stranded carrier DNA and foreign plasmid at 42 degrees Celsius for 40 minutes. So the purpose of adding carrier DNA is what? It blocks the non-specific sites on cell wall and make the plasmid available for uptake. So the non-specific sites on the cell wall are so it will go and attach the cell wall, and it will prevent the non-specific attachment of the plasmid to the cell wall. So if your single-stranded DNA will attach to the cell wall of the yeast, then it will not allow the plasmid to go and attach anywhere on the cell wall. And once you have added the PEG, then it can be easily uptaken in the next step. So another method is the spheroplast method of yeast transformation. Okay. So what is the spheroplast method of yeast transformation? So in the spheroplast method, transformation method, what is done is first the cell wall of the yeast is removed. Okay. So basically the yeast cell is treated with zymolase. Okay. So there is an enzyme. What is the name of this enzyme? The name of the enzyme is zymolase. Okay. Yeast cell is treated with the enzyme zymolase. Zymolase. Okay. So this zymolase, what this enzyme does is it digests the cell wall of the yeast. It will digest the cell wall of the yeast. And then once this cell wall is digested, what you are left with, that is called the spheroplast. So the yeast without the cell wall, that structure is called the spheroplast. And in the next step, what is done? So let's go one by one. So cell wall removal. So the yeast cell wall, which is composed of these different saccharides, carbohydrate molecules. So glucans, manoproteins, chitin. It is enzymatically digested using lytic enzyme zymolase to form spheroplast. Now, the permeability, how the permeability is increased. So the spheroplast lacking the rigid protective cell wall have a fragile plasma membrane only, and it will allow exogenous DNA to enter more readily. Now, in the next step, again, PEG is added. So the PEG will promote membrane fusion and facilitate the uptake of DNA molecules into the spheroplast. And in the final step, there will be this is the regeneration step. So after this DNA is uptaken, then the spheroplasts are incubated in an osmotically stabilized media that prevents osmotic lysis because you have digested the cell wall by treating it with zymolase. After you have done the PEG-mediated fusion and you have to allow recovery the or you have to place it in a media so that the this yeast cell does not burst or there is no osmotic concentration is maintained inside the cell. Okay, osmotically stabilized media sorbitol containing which will prevent osmotic lysis and allow the cell wall to regenerate. It will also so this is the recovery phase as we were doing in bacterial transformation. We were adding the LB media and allowing the bacteria to recover. So same here also we place it in the sorbitol containing medium and allow the cell wall to regenerate. Okay. So the role of zymolase in the spheroplast transformation method. So what was this spheroplast transformation method? Anyone, can you summarize what was this spheroplast transformation? What you need to remember? So you have your yeast cell, right? So yeast cell has a cell wall, isn't it? So that cell wall is digested with is what? It was digested with zymolase. Zymolase. Now once you add the zymolase, then in the next step you add PEG, and in the last step you keep it for regeneration. Okay. So So if you do not treat it with zymolase, then the DNA will not be uptaken efficiently by the yeast cell. That is why we treat it with zymolase. So the answer is what? It is remove the cell wall, isn't it? Zymolase was what is it doing? It was removing the cell wall. It is removing the cell wall. Okay. To partially remove cell walls to produce spheroplast. So you see over here in this figure also, which was there in which was explained to you in the lectures. So this cell wall is gets broken when it is treated with zymolase. Okay, and then you add this peg carrier DNA and other the same thing you do what we were doing in the lithium acetate method. And then so you see here this cell wall has been removed, and now you add the plasmid DNA, and it is easily uptaken by the each cell. Okay. And then you do keep it for regeneration for 4 days. You keep it in the at 30 degrees Celsius. Okay. So for each cell, we usually use 30 degrees Celsius for incubation. Okay. Now then there are methods of what we learned. First we learned about how we can introduce the recombinant DNA in bacterial cell, isn't it? So what were the various methods? One is the heat shock method and another is the electroporation method. Okay, so remember the names, a heat shock and electroporation. Now, in the next couple of slides, we learn that how we can introduce this recombinant DNA in yeast cell. Okay, yeast cell. So there are two methods. One is the lithium acetate method, and another is the spheroplast spheroplast method. Spheroplast method. Now, in the next couple of slides, we will see that how we can deliver the DNA delivery in mammalian cells. Okay, DNA delivery in mammalian cells. So here there are what are the various methods in mammalian cells. So these are the methods. One is this chemical transfection method. Then there is this liposome and lipoplex method. Then bactofaction and another is the transduction. So there are four methods we will learn by which DNA is delivered in mammalian cells. So first is the chemical transfection method. Okay. So the chemical transfection method, we use what? We use lipofactamine. So that is the chemical agent that is used. Um, so, so let's go one by one. So the chemical method of transfection, what it is, what is the basis of it? So the basis of the chemical method of DNA transfection into mammalian cell relies primarily on the interaction between the positively charged chemical reagents and negatively charged nucleic acid, DNA or RNA, followed by cellular uptake, usually via endocytosis. So what is your DNA? Your DNA has the sugar phosphate backbone. As a result, your DNA is what? It is negatively charged positive because of the phosphate group which is present in its backbone. It is negatively charged. So if you have to introduce the DNA inside the bacterial, inside the mammalian cell or any cell, what you have to do is, so inside the mammalian cell, because the lipid, the mammalian cell is also cell membrane is what it has phospholipid. It is yeah, because it will not allow the negative charge molecules to be directly uh, will not allow the negative charge to enter inside the so it is amphipathic, right? The lipid membrane. So it will you have to use some chemical agent so that you can introduce this negatively charged DNA molecule inside the mammalian cell. So what is then positively charged chemicals are used. Okay, positively charged chemical reagents are used. So what are the different chemical reagents are used? One is this calcium phosphate method. Second is the polyplex method, and third is the lipoplex method. Okay, so what is the calcium phosphate? The calcium is what? It is positively charged. Okay, so in this method, what is done is this DNA is mixed with calcium chloride solution and then slowly added to the phosphate buffer saline solution at physiological pH. Now this calcium phosphate. So you are adding calcium chloride to your phosphate PBS phosphate buffered saline, PBS buffer. So you will get this calcium phosphate. Okay, so this calcium phosphate precipitate will form. So the calcium phosphate and DNA forms an insoluble fine precipitate. So the DNA and calcium together, it will form an insoluble fine precipitate. And then this DNA calcium phosphate precipitate, what will it do? It will adhere to the cell membrane and is internalized primarily through endocytosis. So this DNA calcium phosphate precipitate that is formed, that gets adhered to the cell membrane and is internalized through endocytosis. Now the precipitates can also cause the local membrane destabilization, aiding DNA entry into the cytoplasm. So this method is actually not followed nowadays because it has a couple of disadvantages. So because this calcium phosphate, what it can cause toxicity, potential cytotoxicity at high concentration, and it requires a lot of optimization for pH and precipitate formation. So and this method is only suitable for cells which are growing in monolayer. Okay, so there are different types of mammalian cells are have different property. One, some grows as a monolayer, some grows um, as one layer over the other. So there are different types of cells. So once inside the calcium phosphate, so next what is so this precipitate causes local membrane destabilization and aids the DNA entry into the cytoplasm. And once this is entered into, it gets entered into the cytoplasm, this calcium phosphate precipitate dissolves due to low pH, and and the DNA is released into the cytoplasm for nuclear delivery. So what is the entire summary of this method is what you are doing is you are adding calcium chloride to phosphate buffer saline, and as a result a calcium phosphate precipitate is formed. Okay, this calcium phosphate and DNA complex, so a fine precipitate of this DNA calcium phosphate is formed. Okay, so this is complex, this complex is formed. Now then what is done, then this calcium phosphate DNA complex enters into the cell, or what is it is endocytosed by the mammalian cell. Now once it is endocytosed by the mammalian cell, it will combine with the endosome, right? Now once it combines with the endosome, as we have studied, in cell biology that it has low pH. Now at this low pH, this DNA gets released from this calcium phosphate complex, and then it can be integrated in the, or it can be integrated, it moves to the nucleus, and it either can get integrated with the mammalian genome, or it can exist as a plasmid. Okay. So remember the disadvantages. So one of the disadvantages is potential cytotoxicity at high concentration, and it requires a lot of optimization, and it gives very inconsistent results. Okay. So that is why this method is not used as widely as other methods are used. So now this method is used widely. So this polyplex method. So what is this polyplex method? So positively charged polymers such as polyethyl polyethylenimine, PEI, or poly-L-lysine protonate their amine groups under physiological condition and bind electrostatically to the negatively charged DNA. So all these chemical methods, what are they doing? They're basically trying to neutralize the negative charge of the nucleic acid of the DNA molecule. Okay, so here also this polyphenylamiine, what it is doing, it is having a positive charge. So this positive charge and negative charge of the DNA, it will by electrostatic interaction, this negative charge will be neutralized, and then this complex can move inside the cell. Okay, so under and bind electrostatically to the negatively charged DNA, condensing it into compact nanoparticles, which are called polyplexes. So these positively charged polyplexes attach to the negatively charged cell membrane and are taken up by endocytosis. Okay. So now these polyplexes, because they have these NH3 plus groups. Okay, they will have this NH3 plus groups. They will have positive charge, and this will be endocytosed by the mammalian cell. And after that, what happens? The polymers like PI exhibit a proton sponge effect, where buffering action induces osmotic swelling and rupture of endosomes, promoting efficient DNA release into the cytoplasm. Now, in the next step, the DNA is transported to the nucleus for transcription. So this is the polyplex method. Where you are adding a positively charged molecule. So that will electrostatically attach with the negatively charged DNA molecule, and will form nanoparticles, polyplexes, and is uptaken by the cell by endocytosis. Okay. And the next is this lipoplex or liposome based method. So this method is most widely used. Okay. So lipofactamine. Most widely used. So there are cationic lipids like DOTAP, lipofactamine. Lipofactamine, these self-assemble into liposomes or micelles. So when mixed with DNA, they form lipoplexes by uh, by electrostatic interaction between the positively charged lipid head group and negatively charged DNA. So basically, liposome-like structure is formed. Okay. So this positively charged head group, these are positively charged, and this will form a complex with the DNA. DNA is negatively charged because of the phosphate group, and so this will form a complex. Okay, and then this will again be endocytosed by the mammalian cell, and inside this endosome, this low pH will cause the release of this DNA, and now this DNA can move into the nucleus, and and the downstream processes can take place. Okay. This is a liposome-based method. So this is the liposuction reagent. You can see this figure was explained in the lectures. Okay. So the principle behind chemical transfection technique is what? So just now we learned that what it is doing, this chemical transfection, it is neutralizing, or it is yeah, it is the negative charge which is present on the DNA due to the phosphate groups, that is getting the that is the in all these methods, what it is doing, it is neutralizing that negative charge. Okay, so it is coating, basically, that is coating the DNA with polymeric compounds. So these polymeric compounds are positively charged, and as a result, the this DNA or this complex that is getting formed, that can be uptaken by the cell membrane, or but that can be uptaken by the cell by endocytosis. Okay. So the answer is yeah. So here also in this lecture, we we um, we had already learned that the principle behind this chemical transfection technique is what it coats, or it forms a complex with the DNA with a polymeric compound to a or complex the DNA with a polymeric compound to a reasonable size precipitate. So you see here this figure also. So this negative charge and these chemicals are having positive charge. So either you use this polyethylenimine, or you use this lipofactamine, or you use the calcium phosphate that was getting formed. Those are all positively charged. And now this positively charged, and this your membrane is mammalian cell membrane is negatively charged. It has an overall negative charge. So this positive and negative charge will allow the this one to be endocytosed by the mammalian cell membrane, and the DNA to be transferred into the cell. Okay. Now the method involving packing DNA into lipid vesicles for transfection. So answer is what? Answer is option C. So option D, liposome and lipoplex method. So the liposome and lipoplex method, you are using what? DOTAP or lipofactamine. So these were forming. So these are basically they will this is a polar head group. So these are lipid molecules, or they have this polar head groups. It will form a complex with the negative charge which is present on the DNA, and then it will get endocytosed by the cell membrane. So these are basically like detergents only. So pack the DNA in lipid vesicle or liposome. So in this approach, the DNA containing vesicle will be fused with the cell membrane and deliver the DNA to the target cell. The preparation of liposome and encapsulating DNA was a crucial step to achieve good transfection efficiency. So liposome prepared with cationic or neutral lipids. So these are basically what? These are lipofactamine. These are cationic lipids. Cationic, or they can be neutral lipids also. Mainly cationic lipids. So they will form liposome-like structure around the uh, nucleic acid, around the DNA. So this complex that is formed, that is called the lipoplex. Okay, so the DNA and the cationic lipid together, they will form a complex which is called the lipoplex, and and it will allow the complex to be endocytosed. Now bactofaction. What is bactofaction? So bactofaction is basically you are using a bacteria to transfer the genetic material into the mammalian cell, that is bactofaction. So like you must have heard about this agrobacterium tumefaciens. This should be neutralized. Okay, so the bactofaction takes advantage of the natural ability of the soil bacterium agrobacterium tumefaciens to transfer DNA into plant genome. Okay, so as you know that you know that you must have read about the TI plasmid, how this TI plasmid is transferred by the help of this agrobacterium tumefaciens. So in a similar way, these bacteria can invade or be phagocytosed by mammalian cells, and once this bacteria is phagocytosed by mammalian cell, the plasmid can be delivered into the host cytoplasm. Okay. So this method exploits the natural bacterial infection process to mediate intracellular delivery of therapeutic or reporter genes. Now, what is transduction? Transduction means what? So there are three main: transformation, conjugation, and transduction. We learned in class 12. So transduction is when we are using viruses to transfer the gene of interest into the host cell. So that is transduction. Okay. So so you can see this figure here. So what this bacteriophage is doing is it is transferring the DNA from one bacterium into the cell of another bacterium. So you see this is the bacteriophage. It has this tail, and this is the head, and these are the tail fibers. And so what it does, once this bacteriophage will infect the bacteria, it will inject its DNA, its own phage DNA into the bacteria, and then this phage DNA can undergo recombination with the bacterial DNA, and we can or if you have a package, if you are using lambda phage DNA, we have for example, we have this recombinant DNA which is packaged into the head of this phage DNA. Then this recombinant DNA can be transferred into the bacterial cell, and then this by the process of homologous recombination, this recombinant DNA will be integrated into the genome of the bacteria. Okay, so that is what is the transduction process. Okay. So yeah, once inside the bacteria, the what will it do? This virus will undergo, will start assembling its components, and it will start dividing on its own. And then this virus will lyse the bacteria, and it will come out of the bacterial cell. Okay. Now, so here in this figure, you see if this your if you have your DNA of interest cloned into the genome inside the genome of this phage DNA, then this. By the process of recombination, once it has injected its fast genome into the host cell, then by the process of recombinant homologous recombination, this can be integrated into the host genome. Okay. So first step is what? Phage injects DNA. Second step is what? Phage enzyme breaks down the host DNA, isn't it? Now the cell creates so phage enzyme will break down the host DNA, isn't it? It will break down the host DNA, and it will use the cellular machinery of the host organism to synthesize its the different components that it requires, its own enzymes and all other components, these tail fibers and this head and all these, it will try to synthesize. Okay, and now so due to recombination, the DNA, donor DNA will be included. So the viral particles are produced harboring the gene of interest under a suitable promoter. The cells are exposed to viral particles which bind to cell surface receptor and enter via endocytosis or membrane fusion. Now in the next step, the viral genome integration or expression will take place. So depending on the virus type, the DNA integrates stably into the host genome or exists episomally, leading to gene transcription. And in the next step, the inserted gene is translated and functional properties produced. Okay. So the gene transfer method that uses bacteria to deliver DNA into plant cells. So that is what? That is bacterfection, just now we learned. So the gene transformation method that uses bacteria to deliver DNA into plant cells. So it is using the same principle that Agrobacterium tumefaciens uses to inject its plasmid into the plant cell, isn't it? Answer is bacterfection. Okay. Now detecting inserts in the vectors. Okay. So basically, or we can say that screening of host cells. How will you screen for the host cells? Now, once you have transformed the host cell, now there will be some host cells which will have the recombinant DNA cloned in the vector, or it will have the cloned recombinant DNA. Others will have self-ligated vector. Others, there may be some hosts which may not have taken any recombinant DNA. It may not have taken the DNA. Okay, so for that you have to do screening. So there are different methods by which this screening is done. So one is insertional inactivation, then there is reporter gene, then blue white screening, insertional inactivation of CI mutants, and so we will learn all these one by one. Okay, we will quickly we'll have a quick recap of all these concepts which are covered in your lecture. So what is insertional inactivation? So what happens is that, so you see this figure. See this figure. So this is your vector. This is your vector. So this vector has antibiotic resistance gene. It has two antibiotic resistance gene, antibiotic resistance gene 1 and antibiotic resistance gene 2. Okay. Now what we have done, we have, so this antibiotic resistance gene 2 has a multiple cloning site present inside the inside its gene. Okay, it has a multiple cloning site. And what we have done is we have ligated our gene of interest inside within this antibiotic resistance gene too. So you see this figure here. So you can see that this insert has been ligated, or we have cloned the insert within this antibiotic resistance gene too. So what will happen when if you ligate this insert into the it's within the antibiotic resistance gene too? So now because of this insertion, the protein that it was producing, it will no longer be able to produce that protein. Okay, so you have a gene. Okay, you have a gene which is coding for some protein. Now you have cut this gene and you have ligated something else within this DNA. Okay, so this was your DNA, and you have ligated something else within this DNA. Now this DNA, will it be functional? It will no longer be functional, isn't it? So now this bacteria. So for example, if there is a bacteria which has this recombinant DNA, or which has this vector, then then this bacteria can grow in both these antibiotics. So if your media has both antibiotic 1 and antibiotic 2, then this bacteria will be resistant to both antibiotic 1 and antibiotic 2, and it can grow in such kind of media. But if you but once you have but those bacteria which have this plasmid, and then this bacteria can only grow in in media which is supplemented with antibiotic 1. Okay, but it will not grow in media supplemented with antibiotic 2. Okay, so that is insertional inactivation. So the idea of insertional inactivation is that if you are inserting something, it is resulting in inactivation of the gene. So that is insertional inactivation. Okay, so your insertion of your gene of interest is inactivating the the that particular gene. Okay, so a plasmid that has a unique restriction enzyme site within an antibiotic resistant gene can be used to identify those plasmids into which a cloned gene has been inserted successfully. So basically what we can do is that, so the if we have the agar plate, if we have a agar plate, one is supplemented with antibiotic 1 and antibiotic 2, and another we have another agar plate which is only supplemented with antibiotic 1. Then the bacteria which grows in both there will be bacteria which will grow in this kind of plate which have both antibiotic 1 and antibiotic 2, and there will be bacteria which grows in plate which have been supplemented with antibiotic 1, and there will be bacteria which are growing in which are growing in plates which have been supplemented with antibiotic 1, but they are not able to grow in plates which are supplemented with antibiotic 2. Okay, so the bacteria which are able to grow in agar plates which have been supplemented with antibiotic 1, but are not able to grow in agar plates which have been supplemented with antibiotic 2. Those will be our those will be our recombinant. Those will be basically our transformed colonies. Okay, those will be our transformed colonies or recombinant colonies. We can say those are our recombinant colonies. Those are the colonies which we are interested in. Okay. So that is what has been explained here as well. So the plasmid that has a unique restriction site within an antibiotic resistant gene can be used to identify those plasmid into which a clone gene has been inserted successfully. So if the gene of interest is ligated into this restriction site, we have the gene of interest, it has been ligated into this restriction site, the antibiotic resistance gene will no longer be active. Isn't it? It will no longer be active, and consequently, the cells that receive a plasmid without an insert. So there will be few cells which will receive this kind of plasmid. So the cells which receive this kind of plasmid will be resistant to both antibiotics. They will grow in agar LB agar plates which have been supplemented with both the media. But those which receive a plasmid with an insert, so the bacteria which receives this kind of plasmid. So this is one type of plasmid. This is your plasmid 2. So bacteria which have received the plasmid 2 will be resistant only to the first antibiotic, but they will be sensitive to antibiotic 2. Okay. So you have to plate the culture in or plate the bacteria in all the three different media. Media supplemented with antibiotic 1 and antibiotic 2. So in this type of media, both the in this what will happen, which have been supplemented with antibiotic 1. So this type of the bacteria which have received this plasmid, they will grow. Okay. And in the media which have been supplemented with antibiotic 1, what will grow? These will grow, and these will also grow. These will also grow. Okay. So, and you have to again set up another plate where you have supplemented antibiotic 2. Then comparing these, we can say which is our recombinant and which are our non-recombinant colonies. Okay. Because if your media has antibiotic 1, then your both non-recombinant and recombinant both will grow. Here, what you will get, here only your recombinant will grow. Here only your non-recombinant will grow. Here what will happen? Only non-recombinant will grow, isn't it? And here what will happen? Here also, here what will happen? Here your recombinant will grow and your non-recombinant both will grow. Okay. So if you have these two plates also, then also you can identify which is your recombinant colony and which is your non-recombinant colony. Okay. Because if you are agar plate, so this is you have two types of agar plate. One of your agar plates. So this agar plate has both antibiotic 1 plus antibiotic 2, and this agar plate has only antibiotic 1. So the agar plate which has antibiotic 1 and antibiotic 2, in these type of plates only what will grow? Only your non-recombinants will grow. Okay, your recombinants will not grow. They will not be able to grow. Okay, because they are sensitive to A2. But here, what will grow? Here, both your non-recombinants will grow and your recombinant will also grow. So by comparing this and this, you can identify which is your recombinant and which is your non-recombinant. So for example, you have these colonies, for example. Here also you will have these colonies, and along with it, you will have some additional colonies here. So these will be your desired colonies. Okay. So the one is the insertional inactivation. Now next is what we can use reporter genes. I hope you have understood this insertional inactivation. Now reporter genes, what is done? So in reporter genes, so these are genes that are used in genetic analysis because their products are easy to detect and they are often used to report on gene expression. Okay. So one common reporter is the LAGZ gene, which encodes the beta galactoside. So this LAGZ gene, basically it codes for beta galactoside, and this beta galactoside is what it beta galactoside is. Beta galactoside is an enzyme which um, which which uh, which breaks this disaccharide bond. Okay. So if you have lactose, for lactose it breaks down lactose. Beta galactoside is an enzyme which breaks lactose into glucose plus galactose. Okay, it breaks down lactose into glucose and galactose. So it converts a disaccharide into two monosaccharide. Now this beta galactosidase can also act on, so this is its natural substrate, but there are some artificial substrate as well. So it can act on, like this is XGAL. So it can also convert this XGAL into this D-galactose, and it can produce a chromophore substance like this 5-bromo-4-chloro-3-indoxyl. Okay. So this compound it reacts with oxygen in air and it gives a blue color. Okay, so the enzyme beta galactosidase normally cleaves lactose into two monosaccharides, glucose and galactose, but beta galactosidase also cleaves to artificial substrates. So what are these two artificial substrates? One is ONPG and another is XGAL. So this beta galactosidase on reacting with ONPG, it produces the yellow colored compound, and bright yellow substance is produced, but when it reacts with XGAL, so XGAL is basically 5-bromo-4-chloro 3-indolyl galactoside. So once it reacts with XCal, it produces a compound, and this compound, when it reacts with air or oxygen, it gives a blue indigo dye. Okay, so by this you can understand whether. So once you add this substrate in the media, there will be colonies which will turn blue, and there will be colonies which will remain white. So the colonies which have taken up the gene of interest, what will happen in those genes, so we will see what will happen. Okay, so blue white screening we will learn, but understand that there will be some colonies which will be blue, there will be some colonies, and in which case which what will be what we will see now. So what is done basically, the this blue white screening is has has a little concept that you must understand. So in blue white screening, what is done, this beta galactosidase protein is, it is unique. Okay, it is unique. Why? Because it can be expressed as two pieces that come together to form a functional proteins. So this entire, this beta galactosidase protein is a very is a large gene molecule which codes for this beta galactosidase protein. It is around 3000 base pairs. Okay, so we can clone it separately. So there is first 175 amino acids, which the gene which codes for the first 175 amino acids is cloned separately as lag Z alpha, and the remaining is cloned separately as lag Z gene. Okay, so the alpha fragment, this is called the alpha fragment, can be expressed from a plasmid, and the remainder of the beta galactosidase can be expressed from the chromosome. Okay, so so you have this entire gene. Now this entire gene has been broken up. So one part of the gene is one part of this gene is just called the lag Z alpha, that codes for the only the alpha fragment, and the rest of the gene codes for the rest of the lag Z protein. Now when so these two different proteins that have been expressed, they can assemble and give us the active beta galactosidase. Okay, so if the plasmid and the chromosomal gene segments are active, they produce two protein fragments that associate to give an active enzyme. Okay, this is referred to as alpha complementation. So when will the active beta galactosidase be formed? It will only be formed when this alpha fragment comes together with the rest of the lag-Z protein. So the reason for splitting lag-Z between plasmid and host is that the lag-Z gene is unusually large. It is 3000 base pair, and it is the plasmid cannot take up, or the new normal plasmid or the vectors that we use that cannot take up such large base pair, so that this entire lag-Z gene would make the final plasmid too large for cloning. That is why it has been broken into two fragments. The first part is the lag-Z alpha, that is cloned separately, and the remaining of the gene is cloned separately. Lag-Z alpha will code for the alpha fragment, and the other will code for the lag-Z protein. Okay. So you see in this figure also. Now, in order to screen for inserts in a plasmid with the lag-Z alpha gene, a small polylinker or multiple cloning site is inserted into the extreme N-terminal portion of the lag-Z alpha. So what is done? So this lag-Z alpha fragment that is there, that has some that has been engineered in a way to so that it contains multiple cloning site. So what will happen if you clone your gene of interest in this multiple cloning site? Within this multiple cloning site, then what will happen? This lag-Z alpha, the lag-Z alpha fragment, the protein that was getting produced, that will become inactive. That will no longer be able to form, and what will happen? So the active so you see here, so the your insert, you inserted your gene of interest within this multiple cloning site. So as a result, the beta galactosidase that will be produced, that will remain inactive, because your lag Z fragment is not able to will lag Z alpha fragment will not be formed, isn't it? So if the lag Z alpha fragment is not formed, then the rest of the protein that is expressed separately, that will not be an active protein. Okay, but if your lag Z alpha fragment does not have your your gene of interest, then in that case, what will happen? The cells will form blue colonies. But if your laxi alpha, if you have cloned your gene of interest within this laxi alpha, then those will be defective and those will form white colonies. So your white colonies are your recombinant colonies, while your blue colonies are your non-recombinant colonies. So these are your recombinant colonies. Okay. This is what you want. So if you have to screen that which of the colonies have my gene of interest, and which of the colonies do not have my gene of interest, or do not have this recombinant DNA, then you can directly add the substrate and you can see the color. So the colonies which will turn blue, those do have not, those colonies do not have your recombinant DNA plasmid, while those that are white, they have your recombinant DNA plasmid. Okay, these are your non-recombinant for cells. These are your non-recombinant cells. These are your recombinant cells. Okay. So you can go through this text also. Okay. You can go through this text. So this text, you will understand whatever I was explaining. I explained it through the figure. You go through the text. Okay. Now this another is the luciferase reporter system. So this is another again a reporter system. So basically, this is used for checking the expression of a gene. Okay, so if you have a gene of interest, if you have your gene of interest and cloned, you have cloned it downstream of a promoter. Okay, so you have your gene of interest. Okay, you have the promoter gene, and you have fused the luciferase gene along with it. Okay, luciferase gene along with it. Then you can check the expression of this gene by directly measuring the OD actually. Okay, so this the product that is formed that gives a particular luminescence, and you can by taking the intensity of this of this emitted light, you can measure how much your gene is getting expressed. Okay. So basically what is done? So this luciferase is an enzyme that catalyzes a bioluminescent reaction emitting visible light by oxidizing a small molecule substrate called luciferin. So this luciferase is an enzyme that acts on the, what is the substrate of luciferin? It is luciferin. What is the substrate of luciferase? It is luciferin. So this luciferase will catalyze this luciferin, and first of all, what is formed? So this basically this luciferase enzyme has been isolated, or you have cloned the gene which produces this luciferase in a bacteria, and then you are producing this luciferase enzyme. So that is basically the gene which has been taken from this firefly, Photinus spiralis. Remember the name, it is Photinus spiralis. That's the scientific name of the firefly, and this catalyzes the oxidation of d-luciferin. So it will catalyze the d-luciferin, and what is produced? It will produce what? It will produce first. It will produce luciferyl adenylate. Okay, so remember these names: luciferyl adenylate, and then this luciferyl adenylate, and also ATP is also required. Okay, so luciferyl adenylate is produced and pyrophosphate will be produced. Okay, now this luciferyl adenylate, it will react with molecular oxygen, and what is produced? Oxyluciferin. Oxyluciferin is produced. So you see this figure here. So this is your D-luciferin, and this D-luciferin is acted upon by this luciferase, and this ATP is used. So this luciferin is converted into luciferyl adenylate, and then this luciferyl adenylate reacts with oxygen, and we get oxyluciferin. Okay. Now this oxyluciferin then has the ability to emit the light. Okay, this intermediate decays producing oxyluciferin in an electronically excited state, and when this excited oxyluciferin will relax to its ground state, then the energy that is released in the range of visible light, and we can measure the luminescence. Okay, or we can measure the light which is emitted, and we can screen for it. Okay, so the reporter plasmid is constructed when the luciferase gene is placed under the control of a promoter or regulatory element of interest. Okay, so basically your promoter gene, you have your promoter gene, and your the promoter gene is downstream to your promoter gene. You have your luciferase enzyme. Okay, you have your luciferase enzyme. You have your luciferase enzyme, and you can also have your own gene of interest here as well. Okay, so the transcriptional activity of the promoter will drive luciferase mRNA and protein expression. So the amount of luciferase expressed directly. So have your so you see here, so this is your luciferase expressing vector. So in this luciferin expressing vector, you have your promoter and you have the luciferase enzyme. You have the luciferase enzyme, and then when this is expressed, it will. So this so that is what I was explaining. It reflects the transcriptional activity in response to the experimental conditions. Example, presence of a transcription factor, drug treatment, or RNA. So there are different types of reporter genes now used nowadays, like GFP. Also, you can clone the GFP downstream to your gene of interest, uh, and you can see whether your protein is getting expressed, where it is getting localized inside the cell. So such type of experiments are done in we usually we are done regularly. Okay, so that is what is shown here. Eukaryotic cell injection, you lyse the cell, and then you have your luciferin expressing vector, and by measuring the amount of luminescence, you can measure that what is the level of expression of that particular gene. Okay. Now there is another insertional inactivation of CI. So this is done in the viruses. Okay. So basically the lambda phage that we were seeing in the earlier session. So this laboratory strain of lambda, it carries the temperature sensitive mutation in the CI gene. Okay, so this is your lambda prophage, and this is at 30 degrees Celsius, this lambda phage gene will remain integrated within the host genome. Okay, it will remain integrated within the host genome, but when it is incubated at 42 degrees Celsius, so when you it is changed from 30 to 42 degrees Celsius, then this will get excised from the host genome, and then it will start synthesizing its own lambda particles, and it will lyse the cell. So it will get transformed from the lysogenic phase to the lytic phase. So if inactivated by mutation, if inactivated by mutation, then what will happen? This gene is responsible for maintaining the phage in an integrated stage. So this CI gene, this is your CI gene, and this is the gene which is responsible for maintaining the phage in an integrated state within the host genome. Okay, but if this CI gene is inactivated by some mutation, by insertional inactivation, so you have inserted your gene of interest within the CI gene, then the CI gene is no longer active. Okay, and if it is no longer active, then what will happen? Then it will switch to lysis state. Then it will get excised from the host genome, and then this lysis occur. Now, what is done is that in this CI, this is another mutation which this is CI temperature sensitive mutation. So in this temperature sensitive CI mutation, the CI gene is functional at 30 degrees Celsius, at which normal lysogeny can occur. But at 42 degrees Celsius, the CITS gene product does not function properly, and lysogeny cannot be maintained. So at 30 degrees Celsius, there will be this lysogeny is maintained. Lysogeny is maintained means what? This phage DNA will remain integrated within the bacterial genome. But when this bacteria is transferred from the 30 to 42 degrees Celsius, then so this is a temperature sensitive mutation. This it becomes sensitive, and this will get excised from the bacterial genome. Okay, so the culture of E. coli infected with a lambda phage carrying the CITS mutation can therefore be induced to produce extracellular phages by transferring from 30 degrees Celsius to 42 degrees Celsius. Okay, so you have cloned, for example, you have your lambda phage genome, and in this lambda phage DNA, you have cloned your gene of interest. So so you will at first, what will you do? You will grow it at 30 degrees Celsius. You will allow it to grow, and once it has grown sufficiently, now what you will want? You will want the protein to get expressed. So you will transfer it from 30 to 42 degrees Celsius. And at this stage, what will happen? Your gene will excise from the bacterial genome, and now it will start to transcribe, and the protein will start expressing. And your what you get is, because your lambda particles, it will lyse the cell, and it will come out of the bacterial cell. You will get it in the supernatant. Okay. So what is insertional inactivation of lambda CI gene? So you see here that this is a temperature sensitive CI gene. So in this temperature sensitive CI gene, what is done? So when you increase the temperature, your lytic gets converted into. So basically what it is showing, that at lower temperature, when you are lowering the temperature, then your lytic cells is getting converted into lysogenic. So, and in the lysogenic stage, no plaque is formed. No plaque is formed. Okay. But if you have inserted your gene of interest within this CI gene, then what will happen? This CI gene will no longer remain active. So now this CI gene uh, cannot maintain it within maintain its the gene within the bacterial genome, isn't it? It will no longer be able to maintain it within the bacterial genome, and now your lytic will no longer get converted into lysogenic, and it will become lytic now. Okay, the lysogenic state cannot be maintained, and you will get plaques. So the bacterial cell which have uh, which contain which have taken up the lambda DNA, containing your gene of interest. Those bacterial cell you will get a cleared plaque. So these are your recombinant, and where you get turbid plaque, these are your non-recombinant. Okay, did you understand this concept? So this is temperature sensitive CI gene. So when you are inserting your gene of interest within this CI gene, then at it will no longer lower temperature, it will no longer be able to maintain that lysogenic state, and it will turn lytic. And as a result, you will get plaques within the bacterial plate where you are growing. The bacteria will get plaques. Okay. Now, the enzyme responsible for hydrolyzing O-nitrophenyl beta-galactoside in reporter gene assay is what? So just now I said that this beta-galactosidase was produced by what? It was produced by two different genes which have been cloned separately. That is the lag-Z alpha, and another is the remaining lag-Z dash. So this beta-galactosidase can catalyze its normal substrate. Its normal substrate is lactose, or natural substrate is lactose. So lactose will get converted into galactose plus glucose, but it can also catalyze number of artificial substrate. So its artificial substrates are xgal. So when this xgal is broken down, or it is enzymatically catalyzed, then we get a blue colored compound. And when this O-nitrophenol beta galactoside, it can also catalyze ONP. We get a yellow colored product. Okay. So the answer is beta galactoside. Okay. So ONPG, this is O-nitro, this is the structure of O-nitrophenyl galactoside. So this is another artificial substrate of beta-galactosidase. So this beta-galactosidase can break it down into D-galactose and O-nitrophenol. So this, because of this formation of this O-nitrophenol, it gives a bright yellow color. So the colonies will give bright yellow color. Okay. You can apply the concept over here also. So cells which are having this gene of interest within this laxi alpha, those will what? Those will be, those will not be yellow. Those will be white. While those which do not have the recombinant DNA, they will be yellow. So what is the substrate utilized by luciferase? That is luciferin. Okay, that is luciferin. So the same thing. So luciferase, just now I discussed, luciferase will act on D-luciferin. It will convert it into luciferyl adenylate. And then this luciferyl adenylate gets converted into oxyluciferin. oxyluciferin by combining with oxygen. And this oxyluciferin, when it will move from the excited state to ground state, it will emit energy in the visible light range. Okay. Now, antibiotic resistant gene can also act as reporter genes. Okay, how our antibiotic resistant gene can act as reporter gene, similar to what I was saying. So you see this figure. So the bacterial cells which have been transformed with plasmid. So if your plasmid has this antibiotic resistant gene, then so if your bacteria has taken up this plasmid, which is having your antibiotic resistant gene, then what will happen when you treat it with antibiotic? So these type of bacteria will survive. Okay, while the bacteria which have not taken up this plasmid, they will not be able to survive and they will get killed. So when you grow it on an agar plate, which is having your antibiotic in the media, then what will happen? Only those bacteria which have received this plasmid having their antibiotic resistant gene, only they will grow and the others will not grow. Okay. So this is clear. Now remember all these things. So this is again one thing you have some facts which you have to remember. So this ampicillin antibiotic, if it has this ampicillin resistant gene, then what is the gene product of that? It is beta lactamase. If a bacteria has this kanamycin resistant gene, then what is the product? Neomycin phosphotransferase 2. What it does? It causes covalent modification of kanamycin. So if there is tetracycline then resistance gene, then it will code for this ribosomal protection protein. And what is it does is it causes the efflux of tetracycline outside the bacteria. It will expel the tetracycline outside the bacterial cell. Okay. Then there is chloramphenicol. So remember, chloramphenicol produces this chloramphenicol acetyl transferase, which converts chloramphenicol to acetyl chloramphenicol. So once this chloramphenicol is converted into acetyl chloramphenicol, then this chloramphenicol is no longer active and the bacteria will become resistant to chloramphenicol. Okay. So this is remember. So the primary function of chloramphenicol acetyl transferase is what? In reporter gene is that this chloramphenicol acetyl transferase causes acetylation of chloramphenicol. Okay, so remember the table points that were there in the table. Answer is option C. Now we will come to this dideoxy or Sanger sequencing method. This is an important method which I will quickly cover. This dideoxy chain termination method, this is also known as Sanger because it was developed by the scientist Frederick Sanger, or so by this method, so once you have screened everything and you have done all these different method, follow this method. One of the most confirmatory method by which you can which can say for sure that yes, my recombinant DNA has the cloned gene of interest is by doing sequencing. Okay, so ultimately whatever thing you do, that those are not acceptable. Ultimately, you have to do sequencing and you have to show that yes, my sequencing result has shown that my recombinant DNA the has my gene of interest. Okay, so ultimately you have to do sequencing. So when you give it for sequencing, your DNA or plasmid DNA, whatever you have, if you give it for sequencing, then what is done? This Sanger sequencing is usually done. Okay, so why we will see now why it is called chain termination sequence. So what is this method does? We can get the exact order of nucleotides for one segment of DNA. Okay, this procedure provides confirmation of the sequence of plasmid construct created by a researcher. We can get confirmation regarding the mutants. Suppose you have mutated one residue to another. You have created some phenylalanine to say alanine. You have created a mutation. You have changed the codon. Okay, so all these, how will you be confirmed? By sequencing, insertion or deletion of a gene, and can be used for, or it can also be used for sequencing of a whole genome. The approach first involves isolating the DNA sample, and then what is the source of the template DNA can be anything. It can be PCR product, it can be plasmid DNA, it can be genomic DNA, it can be bacterial artificial chromosome, yeast artificial chromosome. So there can be any template DNA. You can give any template. It can be plasmid DNA, it can be other different types of DNA, bacterial artificial chromosome, yak, bag, genomic DNA, anything can be given as the template DNA. Now, what are the components that are required in Sanger sequencing? So, what happens is that first of all, you require a primer, right? So what will happen? The primer will go and bind to your template strand. So during, so this is what happens in normal DNA synthesis. So in normal DNA synthesis, the DNA polymerase will read the template DNA and makes a new complementary strand of DNA. Okay, now to get the DNA synthesis started, a short oligonucleotide primer must anneal to the beginning of the target DNA. So this is your primer and it should go and bind to the start of the DNA, anneal and bind to the start of the DNA. Now, once this is done, the DNA polymerase will come and recognize this primer DNA hybrid. Okay, it will recognize the 3' end of the primer. So this 3' end of the primer is recognized by your DNA polymerase and connects the 5' end of the incoming nucleotide. So you will add this DNTPs. You have the DNTPs in your nucleus, and this the incoming DNTP it will attach the incoming nucleotide to the 3' end of the primer, and hence synthesis occurs in a 5' to 3' direction. So one by one, so if it is T, then D ATP will be attached to this complementary to the thymine. Then guanine for it will be DCTP. So one by one, it will attach the corresponding DNTP. So it will hydrolyze. So what is done here? How this nucleotides are attached? So you can see here, so this is your nucleotide, right? So your nucleotide has your base. Yeah, this is the sugar, and this is your sugar. This is your deoxyribose sugar. Okay, so this deoxyribose sugar, it has a 3' OH group and it has a 2' hydrogen group, and this is your growing DNA chain. Okay, now what is done in this first step? So this is your incoming nucleotide. Okay, this is your incoming nucleotide, and this is the growing DNA chain. This is being catalyzed by the DNA polymerase, and you have your incoming nucleotide. So what this DNA polymerase does is that it first cleaves this gamma and beta phosphate groups that are present, that is first cleaved, and then in the next step, this 5' CH2, which is attached to this phosphate group, that is catalyzed. So this OH group, 3' OH group and the 5' phosphate group is joined together by this DNA polymerase. Okay. And you get, what you get is that this pyrophosphate, this gamma and beta phosphate, these are cleaved, and you also get this proton from this OH group. So the DNA Okay, so the DNA polymerase links nucleotides via phosphodiester bonds. The DNA polymerase breaks the bonds between the first and second phosphates of the incoming deoxynucleoside triphosphate. So this first and second phosphate. This is your first and this is your second phosphate. This gets broken and is then joined to the free 3' hydroxyl of the growing DNA chain. So this is the growing DNA chain will have a 3' free hydroxyl group, and this free 3' hydroxyl end of the growing DNA chain will be joined to your phosphate group, outer two phosphate, and release as well as hydrogen ion. Okay, so this is how the polymerization reaction takes place. Now, what is done in this Sanger sequencing is that, so these are the different sugars that are present. So RNA you have ribose sugar, DNA you have 2' deoxyribose sugar, then there is 2' 3' dideoxyribose sugar. So this 2' 3' dideoxyribose sugar, this is very important because you see here that in this dideoxyribose sugar you do not you do not have the 3' OH which was present in deoxyribose, that is converted into H. Okay, so this OH is now converted into H. So it is dideoxyribose. So this 2' already didn't have an OH group, but along with it, this 3' also doesn't have an OH group. So this is dideoxy. So so what will happen? So if this dideoxyribose comes and gets attached to the growing DNA chain, because this it does not have a 3' OH group, further reaction gets stopped. Okay, so instead of adding the deoxyribose, if my DNA polymerase attaches the dideoxyribose, then my polymerization reaction will stop because this 3' 3' group doesn't have this OH group. The 3' OH group is not present. This is 3' H group. So that is why the reaction will be polymerization reaction will wrong. So the DNA polymerase cannot add another nucleotide to a chain ending in dideoxyribose. because its 3' carbon does not have a hydroxyl group. This 3' carbon does not have a hydroxyl group, so this DNA polymerase cannot add another nucleotide to a chain ending in dideoxyribose. So this is what is utilized in Sanger sequencing. Okay. So in Sanger sequencing, there are four sets of reaction that is done. Okay. So there are four sets of reaction. So in this reaction, you add all of these GATP, DCTP, DGTP, DTTP, and along with that, you add another DDATP you add. Okay. DCTP, DGTP, CTP. In the second reaction also, what you do, you add all these DATP, DTTP, and along with that, you DDTTP also you add. Okay. So, and in the third also, you do the same DATP, DGTP, DCTP, and along with the DDGTP you add. Okay. So so wherever this instead of this thymine getting added, if it goes and if this DDTTP is added, then at moment the reaction will stop. So you will get different lengths of DNA fragments. So this chain termination sequencing, also known as Sanger sequencing, involves synthesizing partial copies of a target DNA that vary in length by one nucleotide and then separating them by size. So you see here, so you are getting different sizes of this DNA fragments. Okay, so for here, this is the DGTP that has been added. So once this DGTP has been added, or D guanosine residue has been added, then this reaction will come to a stop. And then you can read it in the from bottom to top. You can read and nuclear sequence can be identified. And basically, these your nucleotides have been attached to fluorophores, so that you can know which is the nucleotide. Okay. So at the end of a capillary flow electrophoresis tube, record the wavelength of the fluorescence released from the final nucleotide and convert it into the corresponding base. So you see here that there are different DNA fragments that are getting produced. And depending on which fragment is produced, so you pass it through the capillary electrophoresis. So what will capillary electrophoresis do? This capillary electrophoresis has a particular it will separate the molecules based on their size and based on their charge. So based on the size, what will do it do? Based on the size, what will happen? Based on size and charge, this capillary electrophoresis, the shorter fragment, the fragment which is the smallest. The fragment which is the smallest will be eluted first, and the fragment which is the largest will be eluted the last. Okay, so this is not a sec column. This has some different principle. Do not get confused between a sec column and capillary electrophoresis. But what happens in like when you pass it these molecules to the capillary electrophoresis, the one which is having the smallest size, that gets eluted, followed by the larger fragments. Okay, and you can, so this laser light excites fluorescent molecules at the end of the fragment, and the wavelength that is emitted, depending on what is the wavelength emitted, you can understand that what is that molecule, whether it is T or A or G or C. So all these different DDATP, DDGTP, DDCTP, these have been tagged with different fluorophores. Okay. You can read the text. So this figure is taken from this Leninger book. So you can see here, so this is DDNTP analog. So here what is happened? So this 3' carbon is does not have an OH group. It has an hydrogen group. Okay, it has a hydrogen group, and that is why it is called DD, dideoxy. Dideoxy means 2', 3', both are having hydrogen. Carbon both are having hydrogen instead. Okay, here you can see, so that for that, as I was explaining, that you set up four different reaction. So in one of this reaction, you have added, so in all of this, you have these DCTP, DGTP, DATP, DTTP. But in one of the reaction, you have added DDATP. In one of the reaction, you have added DDCTP. In the third reaction, you have added DDGTP. And in the fourth reaction, you have added DDTTP. Now, so what will happen? So when it will get ATP, it will keep on doing the adding the nucleotides one by one. But once it finds once a DDATP is attached, the reaction will stop immediately. So you are getting different fragments. So you see here you have got three different fragments here. So the third fragment, this GDDA, that will be if you run an agarose gel, you will, or a polyacrylamide gel, 5% polyacrylamide gel, then the shortest fragment will be at the bottom. will be at the bottom and the largest fragment will be at the top. So when you go from bottom to top, you can get the sequence of DNA. So it is GATTCGA. So you can do the sequencing in this way. Okay. So you're getting different fragments. So so this is for the example, this is your template DNA. When you are getting G, if you are getting GTP, then this G will be attached. But there can be an instance when you are getting DDGTP. So in that case, a very short DNA fragment will be produced. So for example, you are getting G over here. You are getting G over here. Yeah, then you are getting again a, then T, then C. Then in this C, you are getting a DDGT3. Okay, so it will stop at this instant only. Okay, it will not proceed further. Okay, or you are not getting a DDGTP, then it will continue. Okay, but here you got a DDGTP, then it will stop at this point. So as a result, you will get different DNA fragments, and then those DNA fragments are passed through a capillary electrophoresis, and then you do the sequencing. Okay, so the role of DDNTPs in Sanger sequencing method is what? Sanger sequencing is what? It is terminating the DNA chain elongation. Okay, it will terminate the DNA chain elongation because for DNA polymerase to catalyze the DNA polymerization, it requires a free 3' OH group. Always remember this. It will require a free 3' OH group for polymerizing in the 5' to 3' direction. Okay, so you can read this. Then Then we will move on to the Maxim Gilbert method of DNA sequencing. So this is also known as the chemical cleavage method. Chemical cleavage of DNA at specific bases. So what we are doing here, so there we were using DDNTP analogs, but here what we are doing, we are chemically treating the DNA with different chemicals. Okay, so what is done is basically 4 different reaction is set up. So this is G plus A, C plus T, and C. So this in this G reaction, the DNA is treated with this dimethyl sulfate and piperidine. So what this dimethyl sulfate does is it methylates this guanine residue. This piperidine then cleaves the remaining part of the DNA. Now, if you treat the DNA with formic acid plus piperidine, then what it does, it cleaves. It will cleave at both this G and A. Okay. But if you cleave it with hydrazine, then also it will cleave at both C and T. Okay, it will depurinate both adenine. Depurinate means it will remove that nitrogen base. It will remove the guanine nitrogen base and the adenine nitrogen base. Okay, and if you add this hydrazine, so you need to remember what chemical is doing what. So if it is hydrazine, then it will depurinate or depyrimidinate. So it will remove the cytosine, and thymine is your pyrimidine, right? These are pyrimidine, and guanine, adenine are your purine nitrogenous bases. Okay. Okay, and if you're adding hydrazine plus salt plus piperidine, then it will only cleave the cytosine. After cytosine, it will cleave. Okay, so you see here, so this is in this A plus G. In this A plus G reaction, A plus G reaction, what you are getting? If it is your A plus G, then it will be GCT. Okay, so again, there will be different the fragments you will get. So if it is A plus G, it will cleave after A or G. So it can be either after you can either get a GCT. If it is a depurinating the adenine, then you will get GCT, or it can so happen that it can after this C it is cleaving the G, then you will get this fragment GCTAC, or it can happen that it can cleave after this A, then you will get the entire fragment GCTA CGT, or it can cleave after at the last A. Okay, then you will get this. Similarly, if it's the G reaction, then it will cleave the G. Okay, so it can be PGTAC till this you can get. Okay, or if it is directly cleaving, then P32 will get only. Okay, or if it is a C reaction, then it will cleave after C or C. So you will get only PG1 fragment you will get, and the other fragment you will get what? GCTA. Okay, only this fragment. And then you run it on a gel, and then you read it from bottom to the top, and then you can identify. So your complete the sequence that you get, the complementary to that will be your template strand. Okay, template strand. So you remember this reagents. Okay, this dimethyl sulfate, it will methylate guanine. So this is the G reaction. G methylate the guanine and it will cleave at the G. So it will remove the G. Okay. So till GCTA C. And after this, it will remove the entire the remaining nucleotides will be cleaved. Formic acid, it will depurinate both adenine and guanine. Okay, hydrazine attacks cytosine and thymine with salt. Only cytosine is modified. With salt, only cytosine is modified. But if you are not adding salt, then either C or T can be removed after that. And piperidine is used in all reaction to break the DNA at the chemically modified site. So piperidine is cleaving the DNA at the chemically modified site. Okay. So you read, go through this text. Okay. So it is based on the principle of chemical cleavage, and there is specific susceptibility of nucleotide to certain chemical modifications, creating breaks at defined bases in a radioactively labeled DNA fragment. Now, what is done is that the 5' end of DNA is first radioactively labeled. Okay. Now, the labeled DNA is divided into 4 separate tubes, each containing reagents that cleave at specific nucleotides. So, you take 4 different tubes. So, you said 4 different reaction. One is G, one is G plus A, one is C plus T, and another is C. Okay, another is C. So if the reaction which is G reaction, here what will happen? It will cleave at this nucleotide. It will cleave at G. G plus A means it will cleave at both G and A. C plus T means it will cleave at both C and T. And C means it will only cleave at C. Okay, so what are the different chemicals that are present for G reaction? You add dimethyl sulfate. Dimethyl sulfate methylate guanine. So it will cleave at the G. Okay, A plus G means this what chemical is used? Formic acid. It will depurinate both adenine and guanine. So you will get different DNA fragments. Some DNA fragments will be cleaved at A, some DNA fragments will be cleaved at G. Then C reaction, we are adding hydrazine with salt. Hydrazine plus salt means C, and only hydrazine means C plus T. Okay, so you have to remember these the different chemicals. Okay, and what are the reaction that is caused. That I cannot explain. There is nothing to explain here. I explained already, but this thing you have to remember. Okay. The reagent that is added to replace modified bases and cleave DNA in the Maxim Gilbert method. So that which was the reagent that was cleaving DNA to that is used to replace modified DNA. So which was the reagent that is piperidine. Okay, piperidine was the it is used to replace modified bases and cleave DNA. Okay, so piperidine was cleaving your DNA, isn't it? Isn't it? It was cleaving the DNA. In all these reactions, you have to add piperidine. Along with your whether you are adding formic acid plus piperidine, hydrazine plus piperidine, hydrazine plus salt plus piperidine. So piperidine is cleaving the remaining DNA. Okay. Option B. Now in the Maxim Gilbert method, the formic acid targets what? Formic acid was targeting your purine or pyrimidine. Was it targeting purine or pyrimidine? It was targeting purines. Okay, you go through this. So you see here, purine was formic acid. Formic acid is targeting your purines. Okay, and hydrazine is targeting your pyrimidines. Hydrazine plus salt is targeting cytosine. Remember, you have to remember this. Remember this table. Formic acid. Uh, Answer is option D. Formic acid is targets purine, and hydrazine targets pyrimidine. Okay. And by chemical used in the Maxim Gilbert method to modify guanine, that was your DMS, dimethyl sulfate. Dimethyl sulfate. It attaches the methyl group to your guanine nucleotide and then cleaves at this guanine nucleotide. It will cleave at G. Cleave at G. Okay. Cleave at G. So you see on all this you have added piperidine. Okay. So that was all for today. Okay, so we tried to cover most of the concepts that were discussed in the lecture, and these important. I hope you can now solve the assignment questions. And if you have any doubt in the earlier assignment, then we can discuss those questions also. So thank you for your kind attention, and I'll end today's session if you have no more questions.